Thank you very much. Of course, I would love to be in Paris in person, and maybe that'll happen in some future year, but for now, the remote version will have to do. Thank you very much for the invitation to speak. It's what I've seen so far. It's been really great. I'm sorry that I can't be there in the mornings, but anyway. Before I want to start, I want to acknowledge that here in Ottawa, I live and work on traditional unceded homeland of the Algonquin Nation. In fact, almost everything that we do here in Canada is on traditional unceded homeland of others, and we aim to be more respectful of that, but it's hard often. So what I want to talk about is movement off patch boundaries and movement at patch boundaries. And let's see. There. So here's kind of my little motivational drawing that I have for this. I'm thinking of a single good patch surrounded by maybe not so good patch. And if an individual starts moving around in this good patch, maybe this black line is kind of its movement path. And so it goes back and forth. At some point, it might hit the patch boundary. And that patch boundary, it's got to choose. It can go back into the patch or it can get out of the patch. If it chooses to go out, then all kinds of things can happen. For example, the characteristics of its movement path can change. And so the first question that I want to dig into a little bit is, how can we even describe something like that? That's not entirely new work anymore, so I'm going to remind you of a couple things that people have done sometime long ago. But then the second question that I want to ask is, well, how would that behavior here evolve? If I'm assuming that the interface between the good and the not so good patch is fixed, how would individuals choose to go out or stay inside the good patch, and how would they choose their movement behavior inside and outside of the good patch in order to have something like an optimum? So how would something like this evolve? And then the second question, or the third topic of my talk today, is that some individuals have a third option. They can't just leave the patch or stay in the patch. They can also change the location of the patch. These systems or species are known as ecosystem engineers. And so the third part of my talk will then be what happens if the interface itself can move. But I want to start very simple. First of all, almost all of this talk will be on one-dimensional problems, because those are really the only problems that I understand at this point, or I understand somewhat at this point. And so I want to start very simple and go back all the way to random walks in a homogeneous landscape, because much of what I do will be based on this. So bear with me. For most of you, this will be, I guess, review. But if you have a homogeneous landscape, you can write down very simply an uncorrelated random walk as jumping left and right with a certain probability p. So probability 1 minus p, the individual stays. With probability p, it jumps left or right, and then it jumps in both directions equally. And we can write down a master equation for the location of an individual that's jumping that way. Now, in the appropriate parabolic limit, we get a diffusion equation with a certain diffusion coefficient. And so that's kind of the basic diffusion equation that many of the talks that we've heard here already are using. And that's only one derivation of that, but that's the one that happens to be the way I think of it and the way that's useful in the rest of my talk. So when we get to heterogeneous landscapes, then there are a lot more ways of thinking about this and dealing with it and writing down diffusion equations. One of them that I kind of like a lot is the one that was formalized by Potapov et al. In 2014, but it's not the first reference to that. It's just a really nice paper. So the idea is here that the probability of jumping can depend on the environment anywhere in between the jump off site and the landing site. So that's indicated by this x plus or minus theta delta. And so depending on where the individual kind of senses the environment and decides to jump or not jump, we get a whole family of different ways of writing down the diffusion equation. So for example, what many ecologists like, if the theta is 0, so if the individual measures the environment where it currently is and then decides to jump or not jump, then we get this form of the diffusion equation where the diffusion coefficient, which is now a function that depends on x, is inside both derivatives. If we're thinking of an individual measuring halfway between where it is and where it wants to go, then we have theta equal to a half, and we end up with the diffusion term that's much more known from physics, the kind of Dickian diffusion term. And we can already see that there are differences, and steady states for the two equations look different and all these kinds of things. What is the same for both equations, however, is that if we want to look into nature and see what happens, we would need to measure our diffusion coefficient basically at every single point in space. And so that could be tricky, difficult to do, expensive, or maybe impossible to do. And so what I'm going to do is I'm going to do a slightly different idea where I'm thinking of a heterogeneous landscape to be consistent of or consist of individual patches that are inside homogeneous, but then differ from the outside. So this is pretty much a landscape ecology point of view where people think of patches. And the way I write this down, or also look at this in terms of pictures, is something like this, and something what you saw on my first slide. So these patches can come up naturally, more or less like here. They can be arising from human intervention in agricultural systems. They can arise from human intervention in other systems, harvesting systems. And we typically have these kind of patches. There's still variation within the patch, but for our purposes, we simplify this and say within a patch, we have homogeneous conditions. And so we can write down a diffusion equation inside. And outside, we also have homogeneous conditions. So we can write down another diffusion coefficient with potentially a different d. Then we need to say how we want to couple the two on this red line. And as I said, I'm going to talk mostly about dimension 1. So for me, this red line is just going to be a single point. So what's going to happen at this single point? We somehow need to match the two densities or fluxes, whatever is coming from left and the right. And so here is a little bit of a more schematic figure. We have, let's say, a forest, and inside the forest, we have this movement left and right. And we have a meadow. Inside the meadow, we have individuals moving left and right. And then we have this interface between the two, where individuals, again, have to make decisions. Do they go into the forest or they go into the meadow? And so there is a relatively recent kind of review article that looks at the question of movement in different habitat areas. And so what this one says is that, first of all, the relative movement in different habitat types definitely differs. And this one says in particular that in low quality habitat, movement is higher than in high quality habitat. So if you have a species that prefers the meadow, then they would typically take smaller steps there. And if they don't like the forest so much, then they would take larger steps there. So the blue Ds and the green Ds are typically different. And the other thing that's an example here of a study with butterfly that looked at the alpha. And so typically, the alpha is not equal to a half. So typically, individuals actively choose which habitat they want to go into. And what this here shows is this measures the mobility inside a certain habitat, or the D basically, and the probability of entering that habitat. And so what we're seeing is that if the mobility inside a certain habitat is high, then the probability of entering it is low. We know from here that mobility is typically high in habitats that the animals don't like so much or consider as low quality habitat. And so that means they're not entering low quality habitat very often, they're entering high quality habitat more often. From a research collection point of view, that makes, of course, a lot of sense. You want to be in a place where there's lots of resources. And when there are lots of resources, you want to go slow, that you actually capture all those resources and don't just glance over or run over it. So data says that, yes, all these things do exist, and we need to put those into models. So a little bit more abstracted, and coming back to my first slide on the random walks, I'm now having my line divided into a green part over here with a certain random walk going on, and a blue part over here with another similar random walk going on. The difference is just in the p's. And then here in the middle, I have my interface between the two, and alpha is the probability of going into patch type 1. 1 minus alpha is the probability of going into patch type 2. Now, if you write down the same kind of master equation for the probability, and you carefully make sure that you get all the jumps back and forth in, then you can derive a continuity of flux condition. That's the first one. And that says every individual that leaves this has to enter here, and every individual that leaves here has to enter the green part. So that basically says nobody is lost or gained just from crossing the interface. The second thing that you get is that the density at the interface, so in this case the U here, is in general not continuous. It depends, there are these multiplication factors that include both the diffusion coefficient on each side and also the alpha, so the probability of entering this. There are different ways of deriving these depending on some small different assumptions that I don't want to go into right now. They're all in this paper here that I wrote with a PhD student in 2013. But the main take-home message is that in general, the density does not need to be continuous at an interface, or in general, it is not continuous at an interface. And that may seem surprising at first, but it turns out that it's actually not too surprising, because physicists had figured that out a long time ago. So in physics, you have heat and you have temperature, and heat flows down gradients of temperature. So at steady state, the temperature is constant, but the heat doesn't have to be constant. The heat depends on the material, right? If you put your hand against the wall, at steady state, the temperature will be the same, but the heat is not. And it turns out that even in biology, this thinking isn't new. This goes back to Skellum in 1973. I guess it just was forgotten. So Skellum said similarly, density population doesn't flow down gradients in density, but rather flow down in dynamic level. That's what he termed it. So dynamic level is in some sense the analog to temperature. And so then at steady state, the dynamic level has to be constant or continuous at an interface, but the density does not have to be. There are a number of alternatives of deriving equations for how to couple different patches. One is skew-Brownian motion that was proposed by Cantrell and Kozner in a paper in 1999. And then there is the method that I mentioned earlier by Potapov et al. With these sensing points. These authors didn't put preference in, so the way we put in the alpha, but everything else they did in their examples as well, and they get to very similar equations. Now we can also derive these same kind of matching conditions, the discontinuity as a limit of smooth transitions. So if we assume that the diffusion coefficient and maybe some drift coefficient varies very sharp at that interface, and then we take a limit, we can get to similar matching conditions for the density. So what we have now is we have two or more diffusion equations that are coupled through these interface conditions here. And the first question is, does that even make a difference? Well, this is only one point in your large domain, potentially large domain, so it's a set of measure zero. Does it even make a difference in terms of the results that you get? So the answer is yes, it does make a difference. And here is kind of a landscape, source sink landscape. So source term means the population can grow there. Sink term means that the net growth rate is negative. And these are coupled. And so starting in 1986, Shigesada et al. Looked at models like this to determine spread rates through heterogeneous landscapes. And they had very similar equations like diffusion equations on each patch, and then they had a continuous density matching. So basically, they didn't consider this discontinuity, they just said U on both sides have to be equal. When they did that, they found that the speed as a function of the diffusion rate in the sink patches behaves like this. So small sinks, the faster the individuals move in the sink patch, the faster the population proceeds through the landscape. But when these sinks are large, if individuals diffuse faster in a sink, then the population will eventually get slower and stop altogether. And to me, that sounds a little bit strange. That was actually one of the motivations for the whole work that I'm presenting here. So if you look at this with the discontinuous matching case, then the picture turns into this. So when you have small sinks, you need a certain amount of diffusion in a sink, and then the population can spread. If you have large sinks, then individuals need to move even faster in those sinks to kind of get across before they die. But still, as if they can get across, then the overall population spread rate will increase with the diffusion in the sink patches. So to me, that makes a lot more sense. And we can explain where it comes from. Namely, it comes from this matching condition that we can interpret in terms of individual movement. So now I want to talk about the question of how this movement should evolve at these interface points. So I have a landscape that is, for simplicity, I choose this to be periodic. I have two kinds of patches of lengths L1 and L2. And I have two competing types running in this landscape, and they only differ by their movement parameters. So those are the D's here and the alphas. One type is called U and the other type is called U prime. And the index I stands for patch type 1 or patch type 2. So this is a typical invasion analysis-based study where we're asking which of the two types can exclude the other, which of the two types can invade the other, depending on their choice of the Ds and the alphas. And we did this in two different ways. One of them is we said, let's zoom out, let's homogenize our landscape, and we get to very simple ODE equations that we can analyze completely. And the other one is let's zoom in and just pick kind of one set of one patch of each type, so one blue and one green, and look at the PDE analysis and see how much of that we can continue there. So I'll start with the zoom out part. So on large scale, we want to do homogenization. So we assume that the L1 plus L2, the period of the habitat, is really small. We make a multi-scale approach with respect to epsilon here, and we arrive at an equation for the lowest order term here in our expansion. So for these type of diffusion equations coupled, this is homogenization is of course well developed. The difficulty here was that we had these discontinuities, and so Brian York and Christina Kobold solved that how to do discrete homogenization. How to do homogenization in this context, and so we could apply their method. So you get that the lowest order term U0 is independent of Y, and you get the diffusion coefficient is a harmonic mean of the diffusion coefficients on each patch, and the overall growth rate is an arithmetic mean of the growth rates of each patch. But this arithmetic mean has weights, and these weights contain both the alphas, so the preference of one or the other habitat, and they contain the little d's, so the movement coefficients in each habitat. Because this equation has no spatial heterogeneity anymore. When we just look at resident and invaders, we're looking basically at constant functions, or in other words, we can look at this as an ODE of just the level. And then it turns out that we can write everything in terms of this single equation here, this single quantity here, little k. That's the ratio of the diffusion coefficients multiplied by the ratio of the alphas. So again, alpha was the probability of going into the blue patch. 1 minus alpha is the probability going into the green patch. And we can interpret this in terms of residence times or residence indices. So in a very simple model of diffusion, 1 over the diffusion rate is a measure for a residence index or measure for the time that an individual spends in that patch. If we have this patch preference, so the alphas preferably going in or preferably going out, then we need to change that measure. And it turns out that in this particular case, 1 minus alpha is precisely the right term that you need there. So 1 over the denominator of this is essentially the residence time of an individual in a patch of type 1. And the numerator is 1 over the residence time in a patch of type 2. And in the end, it turns out that this ratio of the residence times is what matters. So the theorem here for this very simple model is that we have an ESS that is a convergent stable strategy, and that is when the ratio of the residence time exactly matches the ratio of the carrying capacities. So individuals should stay in a certain patch precisely according to how many resources there are. So that's kind of nice. And it's a very complete theory, but of course it has this homogenization approach, and we'd like to get rid of this and make this precise on the local level as well. So that's what we did as well. So now I'm looking just at a single pair of patches, and I've just written down all the parameters again. We have the D's are the diffusion coefficients. They differ between the two types. We have these alphas. They can also differ between the two types. But first we look at just one type. And because of the discontinuity at the interface, we needed to prove just a bunch of background theorems like well-posedness of the equation, preservation of positivity, maximum principle, all of the things that you need to apply this theory of mutual invasion. So we did this in this paper in 2019, and it was recently extended to countably many patches and to the study of spreading speeds in a paper by François Mel and a postdoc, no, a doctoral student of his and myself. And I understand that François was there at the conference in Paris on Monday, but can't be here today. So that was kind of the background, basically to show you that the mathematical theory to make this equation work is there. And now we need to analyze it and see what happens in terms of evolution. It really, I'm just looking at the time and I'm stunned by how far I got in time. So I'm sorry. I'll see what I can do here.
I think we've got a little bit more than 10 minutes.
Okay, that's good. So as I said earlier, it's got to do with the ratio of the carrying capacities, and that looks like we're looking at an ideal free distribution. So that means the local fitness at equilibrium should be the same everywhere, and that means the local fitness should be 0 everywhere. And when we do that, because we have constant conditions in each patch, that means on each patch, the density should equal the carrying capacity. And that then determines the jump at the interface. And it determines the jump at the interface precisely with the same k bar that I had in the previous slide. So that gives us the idea that maybe this is the right thing to look at. And it turns out that we can prove this. So the first theorem says that if we have a positive steady state of two strategies, then one of the species, and if one of the species uses this ideal free distribution, then the other must do the same and the densities are equal. So in other words, those are the same species, the same strategies. And the other theorem that we have is that the ideal free strategies invades all the other strategies. We haven't shown, but we are pretty certain that invasion implies fixation. So the ideal free strategy replaces all the others. So in other words, we can prove almost everything that we've proved for the homogenization also on the local scale. So that was the part about the evolution of movement behavior. And now I'm going to go to the second part, or the third part of the talk, where I talk about ecosystem engineers. So ecosystem engineers are species that can change the abiotic environment to make it better for them. So beavers do that. If they don't have enough ponds to put their beaver lodges in, they just dam a river and make a pond, and then they can put their homes there. But plants can do that as well. So this is a Spartina that can slow the flow of the tides and lead to more sedimentation, and that leads to better growing grounds for Spartina itself, and so it will slowly replace the water. And the way we thought of dealing with that is we said we just have binary habitat. But habitat is either engineered and good, or it's non-engineered and bad. And so the good habitat is green, the not so good habitat is blue, and this interface that we've seen before as fixed can now be moved by the species action. So here's again the schematic that I have, and I've added one more thing. So now individuals can go into either habitat or they can stay at this boundary. I can do the same derivation as before. I get a discontinuous density, I get a continuous flux, and you notice that for the flux, I now have this additional term in there that has to do with the movement of this interface. But it just means the flux is continuous. I have a second such equation that looks at the movement of the interface. So the interface can either move, or be moved rather, or it can stand still. And the rate at which that happens, the probability at which that happens, is proportional to the individuals that are physically at that boundary. So when I do this, I get a differential equation for L, and that means I have a free boundary problem that contains the densities of U's on both sides, and it contains a derivative of the L that shows us how much the interface moves. Stefan problems and even two-sided Stefan problems have quite a long history in math biology. Ours is different from all of those in that we have the same species on both sides, so that's kind of biologically different. Mathematically, it's different because in our case, this depends on U and not on the derivative of U. So what can we do with this? Here's again just all the equations in a good habitat, in the bad habitat, the matching condition, discontinuous density condition, and a very general condition for the L that I will specify or have specified earlier. The easiest thing that we can do is we can look at constant speed traveling waves. Because if we do that, then the L equation just becomes a constant here. And the other equation I can simplify to a phase plane analysis, where on one hand, I have just a second order ODE the way we know it, and then on the other hand, I have a boundary condition. And so that makes it relatively easy. So in the case of a Fisher KPP nonlinearity, we can completely determine what the traveling waves are in this context. So this is what it looks like. Here is the traveling wave that we usually look for. So C star is the usual C star, that's the Fisher speed here. That's the lowest speed for which there's a monotone traveling wave. If I go any slower, then I pass here through this line, which means in particular, I pass this boundary line. So that's this condition here, that's a straight line. And so if I pass this, then I have a solution that consists of this part and then this part here. But if C is bigger than C star, then my boundary line is below, and there's no intersection between this and the boundary line. So that means our theorem is that if C is in a certain range that is bounded above by the Fisher speed, then I do get a traveling wave. And I can even get a unique traveling wave that satisfies the boundary condition, the L prime condition for all c between 0 and this Fisher speed. So in other words, this wave is always going to go, well, it's never going to go faster than a Fisher speed on its own. And we can look at how these things depend on these probabilities. So this is the probability to leave a good patch, and the more individuals leave a good patch, the slower the speed. That kind of makes sense because they don't help anymore to push the boundary forward. But with respect to the probability of returning to a good patch, there's an intermediate optimum. If nobody returns to the good patch, then it's just not enough to help push forward the boundary. If everybody returns to the good patch, again, not enough to help push forward the boundary, but at some intermediate probability of returning to the good patch, that's where the speed of the boundary is the fastest. We're currently working on what happens if we put an Lie effect in here. And it's not quite done yet, but one of the things that seems possible is that with an Lie effect, the boundary can be moved faster than in a homogeneous environment. So that's just a little preview of what will be coming eventually. Last slide. I said at the beginning that I will talk only about the one-dimensional case, but that doesn't mean that people have thought about the two-dimensional case before. So Overskyne and Cornell did their original derivation for 2 and more dimensions, and they basically have a perpendicular direction to your boundary in which they have the alpha and the 1 minus alpha going. And recently, Cronin and all studied this and tried to simplify it because this is a really complicated thing to deal with and to derive general biological insights from. So they said, we'll simplify this a little bit. And they got very good matching conditions between these two. There's an old paper by Lindsay and Ward where they looked at asymptotic, really small patches, but they didn't have any alpha in there. So that would be interesting to see what happens. And there's more recent work by Sticky et al. About a domain and a road, or 2 domains and some interchange of individuals between these domains. As far as I know, as far as I understood it, there is no derivation of the corresponding equations from kind of this individual random walk. So it would be interesting to see how patch preference, how the alpha would show up in these equations. And then, of course, we have a whole other challenge because if we have a boundary, then there could be something like border patrols. So individuals may want to stay at the boundary and patrol a good patch or something like that. So we have a whole new way of looking at what needs to be modeled here. That's really future work. With this, I want to thank you all for listening. I want to thank my collaborators, some students and some colleagues who you probably know quite well. And I would like, just like Jen did yesterday, I want to to draw your attention to this upcoming workshop at the Fields Institute. The registration is open now. It will be hybrid to some extent, but there will also be funding available for people who want to attend in person. And of course, we hope that some will attend in person. So I'm encouraging you all to look at this and register there and join us there. Thank you very much.
Hi, thank you for your talk. It was very interesting. Regarding the second part, so you showed how two competitive species interact in such a habitat. Do you know if there are works on predator-prey systems in such environments?
Yes, there are. This is my last slide that I didn't show you earlier. These are all the works that I know of that work with these interface conditions. And so there's one here that talks about pattern formation for predator-prey system in these environments. And it looks at how the probability, the choice of patch, the alphas basically can affect pattern formation. I think that's the only predator-prey work that I know in this context.
Okay, and I have a second question. Can you relate the piecewise constant habitats to smooth habitats in some limit?
we can. So this is this comment down here. If we make the D, so the diffusion coefficient, smooth transition, say something like partition of unity or something, infinitely smooth transition, and then choose the right limit, we can get to at least the steady state equations, if I remember that correctly. We can get precisely these discontinuous flux matching conditions, density matching conditions. That's in the appendix of this paper here.
Okay, thank you.
It's not possible to do the other one by smooth approximation, or no?
The one that has the square roots in it?
Yeah, exactly.
I don't know, and honestly, I don't know. We haven't. I don't think we've tried that. Oh no. So you probably can. If you combine it with this approach, you probably can.
Okay.
I think that's possible, but I haven't done it. Send me an email and I can send you the paper if you want.
Sure, thank you. Any other questions? Adrian, did you have a...
Yeah, hi Frithjof.
Hi, Adrian.
Yeah, it's amazing. You can do the ideal free distribution in heterogeneous habitats. It seems your argument is very general. Like if you have more than two kinds of patches, you can have some way of getting ideal free distribution, right? If you allow different alphas between different patches. And have you thought about?
I think so.
Mm-hmm. Yeah.
So I think that's possible, but I've never thought about it in like with pencil and paper.
Yeah. Have you thought about like time periodic environments, things like that? Since the evolution of dispersal you did in is fairly yeah.
I have not thought of time periodic environments in this context. No.
No, very interesting talk. Thank you.
Yeah, thank you.
If there are no other questions, let's thank Frithjof again.